Good afternoon. Thank you everyone for joining us for today's webinar for Medicenna Therapeutics. Medicenna is listed on the TSX and on the NASDAQ with the ticker Mdna. It's a clinical stage immunotherapy company that uses engineered interleukins that can modulate, fine-tune, or amplify the immune system for treatment of a range of cancers. I've personally been invested in Medicenna for a number of years, and I'm looking forward to hearing the update from CEO Fahar Merchant and CFO Liz Williams. Before we begin, I'll note that the presentation may contain some forward-looking information. You can view those disclaimers on the company's website, and as always, the presentation will be followed by a question and answer period. So if you do have any questions, you can enter them into the Q&A box below or email me at tina@adcap.ca. So with that, I'll let Fahar get started. Thanks for being here. Looking forward to hearing the update.
Thank you, Tina, for inviting Liz and myself for this presentation today. So happy to provide this update to you. So first and foremost, Medicenna, as mentioned by Tina, is dual listed. We were listed on NASDAQ as of last August and also on TSX, the main board, since 2017. So we are headquartered in Toronto. We have, as our last quarter, just over $33 million in cash. Plus we had additional warrant exercises as well as some blocks that were transacted as part of our ATM that sort of brought us to just over $40 million at the beginning of this calendar year. Really nice and clean structure. There is no debt, no preferred shares. And as of our reporting period, just under 53 million shares issued or outstanding, and on a fully diluted basis, just over 61 million shares. So as Tina mentioned, the focus of the company is in the interleukin space that's become very exciting. And we believe we are creating a pipeline of molecules that are best in class, and we'll talk about these today. So first and foremost, we have an exciting program that has finished phase 2 clinical trials, have received very positive feedback from the FDA for our brain tumor drug. This is a drug we call Mdna 55. We'll talk more about it later on. And then we have a series of new molecules that are in preclinical development or soon to enter the clinic for oncology. So the first one I'll talk about today is Mdna 11. It's an interleukin-2 molecule that stimulates the immune system to fight cancer. This has been subject of a couple of very large multi-billion-dollar transactions. I'll mention those later on. And then quite opposite to Mdna 11, we have a second molecule, which we call Mdna 209. And that molecule is designed to dampen the immune system, particularly in patients that have diseases such as multiple sclerosis or type 1 diabetes, where the immune system is overactive. And this again has been a subject of a very large transaction just last week by Merck. And then finally, we have a series of molecules that we call IL-4, IL-13 super antagonists. All these molecules come from Stanford University that Medicenna has exclusive worldwide rights to. And with that compound, we are expecting to provide some exciting new data at a conference coming up in middle of April around a very novel approach that we are using to develop new targeted therapies that stimulate the immune system to fight cancer. So as an overview, let's look at some transactions that have occurred with respect to the interleukin-2 space. So interleukin-2, as I said, has had a large transaction between Nektar, which licensed rights to Bristol Myers. This was a $3.6 billion transaction, which was consummated in early 2018. And then late in 2019, on the right-hand side, we've seen Sanofi's acquisition of another IL-2 company. This company was essentially at a stage where they had just started the phase 1 clinical trial. They were 3 months into the clinical trial, and Sanofi bought them out for $2.5 billion. We will share with you the data with our compound, and actually we are able to show that our molecule, Mdna 11, is superior to both Nektar's therapy as well as Synthoryx's therapy as well. And then last week, we had an announcement by Merck having acquired another very early phase 1 clinical trial company called Pandion Therapeutics. They have their own IL-2, but this IL-2 was for autoimmune disease, etc., not for cancer. And then finally, another company that you see on the bottom right of the screen is a company called Neoleukin Therapeutics. Despite the fact that they are on clinical hold by the FDA, you can see that the market cap is around $500 million, substantially lower than where Medicenna is valued at, and that company is still not in the clinic. So a good landscape for you to understand this particular space. And I will now sort of identify the key milestones that are coming up over the next 12, 18 months for Medicenna. So first and foremost, with Mdna 11, we are expecting to start clinical trials and expect to submit to the regulatory agencies in the middle of this year for us to get permission to start a phase 1/2 clinical trial with Mdna 11 in cancer patients, particularly those with melanoma and kidney cancer. And then in the second half of this year, more towards the end of this year, we would have received safety data, the biomarker results. It will give us a good idea as to how the drug is performing. But more importantly, it'll be for potential partners or potential acquirers of this asset or Medicenna, very much like Synthorx. They started the clinical trial in August 2019. By December 2019, they were purchased by Sanofi. So we will be at more or less the same stage by the end of this year. Also, to supplement and build a pipeline, as I mentioned, we have programs with IL-4, IL-13, different cytokines, but more importantly, a dual or a bispecific cytokine, which will be first in its class, that we expect to identify that compound in the second half of this year, but we will be discussing and presenting some early data at a conference, as I said, in April of this year at the largest cancer conference known as the American Association of Cancer Research. So that should be very exciting, and look forward to sharing those data with you as they come through. And then finally, from a corporate perspective, clearly we have really exciting data with our brain tumor program. And we had a really good feedback from the FDA with respect to getting this drug into the marketplace. But this is obviously a very large program. We feel that this would be best suited for Medicenna to partner or collaborate with a larger pharma that has the resources to take this product to the marketplace. And consistent with our plans with the funding that we have raised to date, we are planning to enhance our management team as well as build out our scientific and clinical advisory board as well. So let's go first to our lead compound, Mdna 55. This is a drug that's, this is a disease, let's put it this way, glioblastoma is by far the most aggressive, uniformly fatal kind of brain cancer. And we would be aware over the past years of Gord Downey, for instance, succumbed to the disease, as well as we had Beau Biden, Senator Ted Kennedy, and Senator John McCain. All of them succumbed to glioblastoma. It's a horrible, very aggressive form of brain cancer. And currently, the patients that are diagnosed with glioblastoma, the first thing happens is that these patients, as would have Gord Downey, undergone surgery. And then after surgery, he would have received a combination of radiation and a chemotherapy drug known as Temodar. And then after 6 weeks of this combination with radiation, they would have then received a high dose of this chemotherapy. And unfortunately, with this, despite this very aggressive chemotherapy, surgery, radiation treatment, these patients will always have the tumor come back. And when it comes back very aggressive. And in a majority of cases, 75% of the cases, the tumors, when they come back, they are deeper in the brain. It's much more difficult to remove them surgically. And at this point, the patients are at the end stage of their lives, and therefore expected weeks or a few months of survival expected in these patients. But what is important just to note one thing about Timodar, this chemotherapy drug. This was a drug that was approved 20 years ago. And today, for newly diagnosed patients, it still is the standard of care. And what Timodar did before it was introduced, as soon as it was launched in the marketplace, what we found is that Timodar improved the survival of these patients that were newly diagnosed, improved the survival by 10 weeks. 10 weeks. And that 10-week improvement in survival made this drug a billion-dollar blockbuster for Merck. So you can see that despite the fact that just 10 weeks of improvement in relatively healthier, newly diagnosed patients, you can see how much of an unmet need there is. Our focus, although we are pursuing, and I'll share data with you in patients that have relapsed, the bigger upside for the company will also be in treating patients that are newly diagnosed. So let's talk more about how this drug works. So Mdna 55 is, if you can see the cartoon on the right-hand side of this slide, the diamond-shaped structure is a molecule known as interleukin-4. Now, interleukin-4 actually is a nutrient. It makes tumor cells grow much faster. So what the scientists at the FDA did was they took that growth factor, namely interleukin-4, and attached to it a very powerful payload that is going to kill the cancer cell. So in essence, created what I would say a molecular Trojan horse, sort of tricking the cancer cell to take interleukin-4, thinking that it's taking a nutrient for it to grow, but actually we are delivering this very powerful toxin inside the cell and killing it. So this is the drug and how it works. But what is really important to note is this particular toxin. It might sound a potent toxin, but it's very targeted. It's very selective. The thing is this, that healthy brain does not have the receptor known as the IL-4 receptor. You need the IL-4 receptor for interleukin-4 and the toxin and the drug to be delivered into the tumor. And because healthy brain doesn't have this biomarker, the healthy brain is essentially safe. The second thing about glioblastoma, and the reason why it is such a difficult tumor to treat, is that there is a blood-brain barrier, and drugs that are taken locally, they simply will not cross the blood-brain barrier. So if you take a drug in a pill format, that drug doesn't get to the tumor, and therefore these drugs generally don't work. But with the Mdna 55, what we do is we use the same simple technique that neurosurgeons use all the time, namely that of conducting a brain tumor biopsy, or for that matter, any kind of biopsy in the brain. It's a routine procedure. That's the first thing neurosurgeons are taught when they are trained to be neurosurgeons. So it's essentially a similar technique where the catheter is inserted into the tumor, just like a biopsy needle would be placed in the tumor, and the drug is delivered. It's a single one-time injection of the drug directly in the tumor. This way, you bypass the blood-brain barrier. You do not end up with all sorts of toxicities that you find with chemotherapies because you take them orally and you affect your liver, the kidney, you have hair loss, etc. That is completely avoided. And then finally, one thing that is very unique about Mdna 55 is that it destroys a shield that targets, that protects the tumor from the immune system. So let me give an example. If you think of a B-52 bomber, that bomber has a coating on the surface of the aircraft so that the radar cannot find the aircraft. The same thing happens in brain cancer, is that they have what we call a tumor microenvironment. It's a shield that surrounds the tumor, and basically the immune system cannot see it, and therefore That's why most drugs don't work in these patients. So the thing about Mdna55 is that it targets the shield. It actually destroys the coating or the shield that's hiding the tumor from the immune system. So the great thing about Mdna55 is that it not only destroys the protection around the tumor, it also destroys the tumor at the same time. So you've got this 1-2 punch that allows this drug to show such impressive results that I'll share with you now. So when we look at patients that are treated with these drugs, this is what you would normally see in a patient. So on the left, on the top left, you can see the circled area. That is where the tumor is located. And right underneath, you can see a colored image where the blue part really is an actively growing tumor. The red portion really implies that the tumor cells there are dead. And as you go from the left to the right, you can see that by day 60, there is less blue. And by day 120, namely 4 months, most of the blue has disappeared and replaced by the red structure that you see there. And that is because it's just a single treatment of the drug. Once we inject the drug, the immune system takes over and starts destroying the tumor. So this is very exciting. And if you look at what we see in these patients' tumors and how they shrink, on the left-hand side, what you see here is a bar chart. So the blue show that the patients did not benefit from treatment. The tumors continued to grow, but that was just 6 out of 32 patients. The 26 remaining out of 32 patients, you can see that either the tumor stopped growing, these are the green bars, or continued to shrink. So we had more than 80% of the patients in this clinical trial where the tumor stopped growing or started to shrink. So that was really exciting. Typically, you wouldn't expect more than 5 to 10% of current therapies to show this kind of effect. And then on the right-hand side, what you see is a survival curve. So you can see that blue plot there that shows what percentage of patients are alive at different time periods. And you can see that the median survival of patients that received a single treatment with Mdna55, this is after relapse. In fact, some patients, 20% of the patients had a second relapse before they received the drug Mdna55. You can see median survival there at 15.7 months. Now, what we did was we then conducted a separate clinical trial where we enrolled patients that had received other therapies that are currently approved. So we identified exactly the same kind of patients that were treated in Medicenna's clinical trial. And when we looked at their survival was, as you can see, just 7.2 months. So with one treatment, with Mdna55, we are seeing that we are more than doubling survival. We are improving survival by 8.5 months, not 10 weeks, like Timodar got approval in newly diagnosed patients. This is 8.5 months, doubling of survival in patients that were end-stage recurrent patients. So this was really encouraging. And when we look at the results with other products that are in the marketplace, temozolomide or TMZ, for instance, median survival there in recurrent GBM is only 5.4 months. And then when you look at Avastin, which is a drug from Roche in combination with another chemotherapy agent, lomustine, you can see median survival about 9 months with that combination approach. Whereas on the extreme right, you can see with Mdna55, we are able to achieve median survival of 14 to 15 months. And in a subgroup of patients that received a low dose of Avastin, you can see median survival approaching 19 months. So clearly the data are very promising. We are seeing very considerable improvement in survival in these patients. And as a result of this data, we had an end of phase 2 meeting with the FDA. And here we received quite considerable concessions from the FDA in designing a novel clinical trial. This is the first time the FDA has ever approved this kind of clinical trial where the majority of patients, typically in phase 3 clinical trial, you treat half of the patients with the drug and another half of the patients get whatever is approved or standard of care. And what the FDA said to us was we would be allowed to enroll majority of patients in our control arm from actually a database. We would not have to enroll patients and ask them to go under treatment, except for 1 in every 3 patients, or 1 out of 4 patients would get the standard of care. 3 patients would get our drug, but the remaining 2/3 of the patients would be from a database. And these patients would have been received treatment with our drug anytime since 1st of January 2016. So this was the first time the FDA has done this. This allows us or our partner to conduct the clinical trial, which is less expensive. It expedites the clinical trial. It gets a product to the marketplace much more quicker. And in the end, it costs a lot less to conduct a phase 3 clinical trial. So that was very encouraging. And then what we can see here, what we are looking to do with Mdna 55 is to partner this drug. We are hoping to enter into a collaboration middle of this year. This would allow the partner to not only pursue recurrent GBM, but also newly diagnosed glioblastoma patients who are much more healthier, patients that have lung cancer or breast cancer where the tumor is spread to the brain. That is a really good patient population. So you can see the market opportunity here with Mdna 55 for brain cancer alone is about $2 billion. If we now look at other tumors outside the brain, what we found is that these other tumors, there are about 20 other types of cancers that have the same IL-4 receptor, the biomarker where Mdna 55 targets. And you can see that these patients are a huge market opportunity. In fact, if you look at IL-4 receptor patients that are diagnosed with cancer every year, there are 1 million new cases with this kind of biomarkers. So the opportunity for Mdna 55 is great, and we're hoping that we will be able to partner this program so a bigger pharma can take this program not only for GBM, but other cancers as well. And then finally, I will say a few words about Mdna 11. This is a drug which is an interleukin-2 drug. This drug has been, in fact, was the first immunotherapy drug ever approved for treatment of cancer, specifically kidney cancer and skin cancer. This is a drug known as Proleukin. It's still available in the marketplace today. This drug has been able to show that with treatment of patients with kidney or skin cancer, about 5 to 10% of these patients have complete remission, meaning that the tumor never shows up ever again. So it has this distinct ability to treat these end-stage patients and cure them. But the problem is that this drug is incredibly unsafe. It is so toxic that patients actually have to be in intensive care unit to be treated with this drug because the side effects are life-threatening. And the second thing is that because this drug is a rather small-sized molecule, it clears through the bloodstream and the kidneys very quickly. So patients have to be dosed literally every 8 hours continuously for 9 days. So with that in mind, what has happened is companies like Nektar, like Synthoryx, etc., have gone out there and taken Proleukin and created versions of this, what they call next generation IL-2. But what happens is that those molecules, although they are safer and they stay in the blood much longer, the problem is that those drugs have lower efficacy. They are not as potent. And so far, we have not seen any patient with Nektar's drug or Synthoryx's drug to show tumor shrinkage when it is used on its own. The second thing is that these patients, when they create this next generation IL-2, the manufacturing process is very complex, and that in turn causes situations from a regulatory perspective, stopping and starting clinical trials because of manufacturing issues. These are not issues with Medicenna's approach. We have created a drug, Mdna 11, that is long-acting, that is safe, and has shown considerable improvement in efficacy of this drug. So here we are comparing our drug in an animal model. This is a colon cancer model in mice. And we're comparing with Nektar's drug, which is the drug that's been licensed with Bristol Myers. So on this graph on the left-hand side, we're combining Mdna 11 with a checkpoint inhibitor. And that orange plot that you see on the graph shows that all these mice are tumor-free. The tumor doesn't grow in these mice. And we kept the study going for, believe it or not, 200 days. That's sort of equivalent to about several years in a human being's life, with just 2 treatments lasting 2 weeks. So these were 2 treatments, 1 week apart. That's it. And then if you look on the right-hand side is Nektar's drug. This drug was administered 3 times for nearly about 3 weeks. But as soon as the treatment was stopped, as you can see, the bottom plot, the tumor starts to grow back up again. Not like the way you see with Mdna 11, where the tumor stays completely under control. But what is really more exciting is when we look at some additional data. So if you look at the top portion of this slide and the third graph from the left, you can see that we have treated mice with just Mdna 11 alone. And what we found is 5 out of these 8 mice were tumor-free. There was no tumor in these mice at all. But what we did was we then challenged, we injected more tumor in these mice at different times. You can see right underneath the 3 different arrows to show you that we injected the mice. We challenged 1, 2, and 3. And despite the fact that we challenged these mice, the tumor never showed up, implying that somehow treatment with Mdna 11 had created some kind of a vaccine-like effect where the tumor would never show up and the mouse or the mice would be essentially free of cancer despite the fact that you are injecting more tumor cells. And then on the extreme right, when you combine it with a checkpoint inhibitor, you can see 100% of the mice are tumor-free, and that even with the fact that you keep on challenging these mice multiple times. So this was really encouraging data that we were really excited with, and therefore decided to pursue studies with non-human primates. So this is studies in monkeys. And on the top, what you see is the stimulation of immune cells and how these immune cells are proliferating, how they are growing following a treatment with Mdna 11. So on the left, as you can see, as we increase the dose from 0.01 all the way to 0.6, we get about 80 to 90% of the cancer-fighting T cells to be completely activated and attacking, to be designed to attack cancer. And that effect you can see lasts for nearly 2 weeks before it gets back to baseline. Now, if you look at the same treatment or the same kind of clinical or study done by Thor 707, which is Sanofi's drug, which, as I mentioned to you earlier, was acquired. Sanofi purchased Thor or Synthoryx for $2.5 billion. You can see that in monkeys, they need to dose monkeys every week, every 7 days to get the same or similar kind of effect. So you're seeing a much better efficacy with Mdna 11 when you compare it with Thor 707. And as I showed you earlier in the mouse studies, you can show that Mdna 11 works better than Nektar's drug, NKTR-214. So on both fronts, we have a candidate molecule that we feel is best in class, interleukin-2. As you can see in this slide, there have been multiple transactions with Nektar, with Synthoryx, and more recently in the autoimmune disease space, Pandion being acquired by Merck for just under $2 billion. And finally, another company, Neoleukin, which is currently on clinical hold, you can see that their market cap is about $500 million US, clearly showing that Mdna 11 and Medicenna's valuation is undervalued right now, and we look forward to sharing more news in the coming quarters and hopefully have a molecule that is best in class, not only with respect to the IL-2 program, but also for the brain tumor program. So with that, I will take calls and be happy to answer any questions. Just to round up here, just to let you know that key highlights with respect to Mdna 11 is that we will start and submit a clinical package to the regulators middle of this year. We'll announce key data on the safety, but biomarker data by the end of 2021, and then demonstrate efficacy data the following year. And then on the left-hand side, what you see is our ability to build a big pipeline around our Superkine platform. And we will be announcing some new data on a next generation IL-2, IL-13 bispecific Superkine at the next conference, which is the AACR conference on April 15th. This is an approach where we believe where tumors are not responding to checkpoint inhibitors, we believe a bispecific cytokine or superkine, such as the one that we have designed, will make it much better for us to treat more aggressive and difficult tumors like pancreatic cancer or prostate cancer, etc. Okay, so thank you very much, and Liz and myself are available for any Q&A that you might have.
Yeah, thanks, Fahar. And as someone in the audience mentioned, it's International Women's Day today, so happy to have a female CEO on board as well. We've already been getting a couple questions from the audience, but why don't we start with Mdna 55? Obviously, some really significant data. Can you tell us if there was any specific rationale behind getting a fast track with the FDA, or was it just generally seeing these overall survival data obviously very significant?
Yes, I think the sort of two things here. Of course, the drug already has fast track designation. It also has orphan drug designation from the FDA, as well as the European Medicines Agency as well. The key thing here is there are two things. It's not only tumor shrinkage that is important. That's what the FDA likes to see. But more importantly, we have seen that in the past that sometimes just because a tumor shrinks does not translate necessarily into significant survival benefit. And this was very important for us to demonstrate not only tumor shrinkage, but more importantly, the survival improvement that we were able to show the FDA. So those are the two important things. And by and large, the gold standard for the FDA is survival benefit. And that's what we've been able to show.
And you've mentioned a couple times Mdna 55 is a single treatment. And have you done much exploration on the side of, or any work behind potentially doing more than one dosage?
We have had a handful of patients that were treated in previous clinical trials, including patients that were administered Mdna 55 on a compassionate basis through a second treatment. We did see benefits in those patients that received a second treatment. So if the tumor comes back, there is potential for further extending the life of these patients. But the protocol was set up so that only patients would receive one dose. The other thing that was quite interesting we found was that when you treat patients with brain tumors, whether it's by radiation and also with immunotherapies like Mdna 55, there is a tendency for the tumor to inflame, and there is sort of edema, etc., that takes place in these patients. And to control those inflammation, what happens is that patients routinely with brain cancer get treated with steroids. And one of the things we did in some of the patients that received the high dose is used a protocol that's to avoid steroid use. They use a drug known as Avastin, which is Roche's drug. And we used that drug at an extremely low dose and just for 2 or 3 times in these patients. And what we found is in this small group of patients, the median survival was 21 months, which is nearly 3 times longer than what you would expect with standard of care. So it seems like it's not only the benefit potential for repeating the treatment with another treatment with Mdna 55, perhaps 6 months or a year later, but the potential to avoid using steroids, because steroids have a whole sorts of side effects. It's better to limit use of steroids. But if you use low dose Avastin, we are seeing quite a dramatic improvement. So there's an opportunity to not only second dose with Mdna 55, but also combine it with other therapies.
And you mentioned partnership discussions, and that's a couple members of the audience had some questions about that. Are they interested in those opportunities as well, or are they purely focused on recurrent glioblastoma? And there's some questions about kind of timing around potential partnerships as well.
Right. So indeed, one of the reasons why we feel that this drug needs to be partnered is that the best outcome for these patients would not only be for recurrent GBM, but newly diagnosed patients. So when patients are first diagnosed, they are healthier and they have a much better immune system and therefore can better fight the cancer. So by partnering, we would expect that a partner, and one of the key things that we are looking in a partner is not only to pursue and commercialize the drug for recurrent GBM, we want them to pursue a newly diagnosed glioblastoma. We want them to also look at metastatic brain tumors and also pediatric gliomas, etc. So we think the opportunity for this drug is multifaceted. There is basically a pipeline within a product. So that's one thing we're looking for a potential partner. The second thing with respect to timing is that we are sort of trying to, having had a really clear pathway with the FDA, the next step for us has been to identify what the pricing will be for this drug. Can patients get reimbursed for this therapy? These kind of analysis have just recently been completed. And also what the market opportunity is by interviewing neurosurgeons and neuro-oncologists. So all this data has been collected. We have set up our data room and provided access to a number of different companies who are currently reviewing these materials. We are hoping that we would get some kind of a term sheet or a non-binding term sheet, hopefully by the middle of this year, that could then transpire into a definitive agreement soon thereafter. So the timing is we're hoping for a second, third quarter of this year for something to be definitive. Okay.
And are discussions mostly U.S. focused, or are you looking for a global partner on this?
We are looking both. We're looking at regional companies as well as companies that are interested in ex-US markets, including Asia, or even a global entity that might be interested in global rights. So it's quite open right now.
Okay, and I'm not sure how much you can get into this, but is there a possibility of spinning out Mdna 55 from Mdna 11, effectively having two separate deals?
Sorry. Well, I mean, that's a possibility. I think that's something that could be contemplated. I think we certainly, as a company, will see if there is interest. Clearly, that makes, in a sense, our Medicenna shareholders would be able to get a much bigger slice of the cake if we keep it within a spin-off entity. But that will depend on the pros and cons of those two different options. So we're not keeping it, that option is open to us. Clearly, we'll see which one is a better way to pursue.
I think, just to add, we do anticipate it'll be two separate transactions, likely a partnership for Mdna 55 and a separate transaction around Mdna 11, or someone willing to pay a lot more for both.
Okay, and then As we're talking about partnerships, are you looking to add anyone to the team in that regard? There's a comment that seems to be more drugs in the pipeline that you keep discovering. So curious to know if you're looking for new team members or to add in the business development side of things.
Yeah, so we are looking at that. That search is currently underway. We are currently working with consultants, but we'll be bringing in individuals on a full-time basis into Medicenna, as well as building our scientific team. As you can imagine, we are looking to bring 3 VP level scientists into Medicenna, as well as CSO and CMO. So these are more difficult hires at the C level, but we are currently in search of those candidates that would support building our pipeline and also partnering those assets as they mature.
Okay, great. And we had a couple of questions on IL-2 as well. Can you provide some color on the advantage of genetically fusing sequence for the IL-2 superkine to albumin versus using pegylation?
Right. So, I think that's a really good question. I think that's something that pegylation, just for the benefit of everyone, companies like Synthorx, Nectar, as well as Neoleukin use pegylation. And what pegylation is really polyethylene glycol, a key component of your antifreeze that you would use. But that approach is basically you take your IL-2, which is a protein, and you chemically link it to a plastic. And by doing so, the challenge is this, that the plastic could bind to any of the different amino acids on the protein. So you end up with a molecule. Every time you make a batch of this drug, yes, you end up with a pegylated molecule, but every batch might have different number of polyethylene glycols per protein. So in some cases, you might just have 1 peg. In other batches, you might have 2 to 5 or 6 pegs, for instance. So you end up with a molecule which every time you make a batch, it's a mixture of different amounts of polyethylene glycol on your interleukin-2. That's the problem. That's one of the reasons that Nectar in 2019 announced that their phase 3 and phase 2 clinical trials had batches of materials that were not consistent from batch to batch, because pegylation is just that. You never end up with the same kind of molecule every time. Also, we saw some issues with neoleukin being put on clinical hold. Whether that was because of pegylation or not, I'm not sure, but nevertheless, it's more difficult to determine the content of your protein. With genetic fusion, the great thing is that you take the entire genetic sequence of our IL-2 superkine or IL-4 or IL-13 superkine, and you know the genetic sequence of human albumin, that's well known. You put the entire gene sequence and tie it up together in one long sentence. Just think of a long sentence with letters connected to each other, and you insert the entire gene sequence into your microorganism or a cell line that's going to produce a protein. So every time you end up with the molecule Mdna 11 or any other albumin fusion protein, you always have the same ratio. It's always 1 IL-2 superkine with 1 molecule of albumin consistently. Batch to batch to batch, you end up with the same protein, and therefore you don't have that concern about pegylation. The other big advantage of albumin is that albumin tends to accumulate in tumors. So by having a molecule like ours, IL-2, fused to albumin, what will happen is the drug, Mdna 11, will end up where the action is. That's where the drug is needed the most, and albumin will take it right to the tumor, and therefore you have that other big advantage which you don't have with pegylation. I hope that answers it.
We had another question about the safety profile for Mdna 11. Can you comment? Obviously, that's an important value added for Medicenna. Can you comment on why you think it's safe in humans at this stage of development?
Well, it's always difficult to predict safety, but I think the key data that's important before you start human clinical trials is the data that you generate in non-human primates. And we know that the non-human primates have IL-2 receptors that are very similar to human IL-2 receptors. And to date, what we've done is, if you just look at Nectar Therapeutics, their drug, they call it BEMPEG, which is a pegylated IL-2. The current dose that they use in human clinical trials, and maximum tolerated dose, is 6 micrograms per kilogram of patient body weight. Okay, 6 micrograms. In our non-human primates, we started at 10 micrograms and increased it all the way to 600 micrograms per kilogram without having to see issues such as vascular leak syndrome or hypotension. These are kind of side effects that you see with interleukin or proleukin. So that was very encouraging. So this is at least a good sign that, we've got a nice wide therapeutic window, and we've seen activity of the drug as low as 10, 30, 100 micrograms per kilogram. And even at 300 and 600 micrograms, we haven't seen signs of immune suppression, or rather vascular leak. So that's giving us confidence that this drug is relatively safe, and we can treat patients every 2 weeks. And finally, when we had a meeting with the regulators at the MHRA, they actually encouraged us to enroll patients right at the therapeutic dose. They want to make sure that the very first cohort of patients, because when you do a safety portion of the clinical trial, you start at the lowest dose and you gradually ramp up the dose until you reach the maximum safe dose. And the regulators in the UK basically said that they would encourage us to start treating patients right at the beginning, in the very first group of patients, at a dose that is going to be therapeutically beneficial to the patient. So it gives us confidence that the regulators have looked at it. They've not come to us and said, we want to go and start at sort of some homeopathic dose before we treat patients at higher doses. But those data, together with the monkey data, give us confidence from a safety perspective.
So in discussing the primate data and the animal trials that you highlighted, they were reinfected over time. In comparison, do you have a sense of what the degree and speed of mortality would be on an untreated cohort?
Well, I mean, the thing is, these monkeys, by the way, in this monkey study, they did not have a cancer, so these were healthy monkeys. So we were just looking at safe dose, and that's basically it. And we did not see any mortality in any of the animals we've treated so far, any of the monkeys we've treated so far at doses as high as 600 micrograms. However, in the mouse studies, what you can see here is, if you look at this plot here, when you don't treat these mice with a drug, and they're simply receiving vehicle or saline, you can see the plot here on the black. That's where within 25 days, all the mice by day 30 are dead, and that's it. So the survival of these mice is very poor. In fact, even with the checkpoint inhibitor, 2 treatments, you can see that these mice are not doing well. Whereas with Mdna 11, the blue plot, you can see that the tumor is growing but stabilizes over a period of time. And then when you combine it with a checkpoint inhibitor, you can see 100% of the mice are tumor-free. So that's a big difference here. So these mice would normally die if they did not receive any treatment.
And there's another question from the audience. I think you mentioned an acquisition that was done mid-trial. Has that company had a readout yet for that?
Right. So the company that was bought out was Synthorx. That was a mid-trial phase 1 acquisition by Sanofi for 2.5 billion. So far, they have reported only phase 1 data and dose escalation data last November. We haven't seen any new data yet, but at that point, they did not show if any of the patients had tumor shrinkage. So call it partial or complete response was not observed. But of course, they continue to escalate the dose and using combination strategies. We are hoping that they will report more results, maybe at ASCO or at the conference next month, or maybe even at ASCO, which will be in May, June of this year. So nothing yet that we can share that we are aware.
Okay. And any comment on whether or not there's a chance that the IND will not be approved, and what will the study's size be, and expected timing on the start?
Sorry, Tina, you're breaking up.
Is there a chance that the IND will not be approved, and what would the timeline be to start?
Well, I think it's something that's not in our control. Of course, it's a regulatory question. We cannot say whether it will be approved or not, but we have the opportunity of seeing what are the issues that have resulted in the IND not being approved, and we have made sure that the concerns that were issued with respect to neoleukin, those are not our concerns, and then we've addressed those issues. So we are not expecting any problems with that kind of analysis or any deficiency from that perspective.
Okay, and any reason why COVID may delay some of these trials? Or are you expecting everything to be as normal?
Well, the thing is this, that of course, one of the things we are looking at is conducting the clinical trials in UK and Australia. As you know, COVID vaccination is undergoing a pretty impressive progress in the UK, as well as there's been a very low incidence of COVID in Australia and New Zealand. So we'll be looking at starting the clinical trials there first before moving to North America. So fingers crossed, we are hoping that assuming everything stays as they are today and the progress continues with respect to vaccinations and incidence rates stay at the low level in Australia, we should be on track.
Okay, great. And we have a couple of financial questions, so why don't we turn to that? Let's talk about your cash runway. I believe in the last quarter you had cash and said you had cash until the end of 2022.
That's correct.
Yes. Yeah.
Okay.
Yeah.
And you had announced this ATM facility as well. So can you give us color on why you decided to launch an ATM, where you're at in that process?
Yeah, so the ATM was filed December 31st. So we have the ability to turn it on or turn it off as we choose. And the primary reason for setting it up was it gives us the opportunity to take advantage of, positive news, high volume stock days, just to add to our balance sheet. It also, we've been very active marketing, talking to new institutional investors. Often they want to take an initial kind of starter investment of a couple 100,000 shares. And so the ATM gives us the ability to get them into the stock. I know there's a lot of speculation about having it on. It is not on at this price level. So we are price sensitive. We do believe the company is undervalued. We did report in our quarter that we had used the ATM in January, and we did place a couple of blocks of shares with institutions, but it hasn't been on actually since January. So yeah, we're very careful about when it's used and if we'll use it. So it's very common for NASDAQ listed companies to have an ATM in place. It's a tool in your financial toolbox to have available when needed, but it doesn't necessarily mean that it will be used.
And are you able to comment on the pricing level at which you would consider using it? And then would the cash runway include a potential deal with Mdna 55?
So the cash runway does not include a potential deal with Mdna 55. And in terms of price, I mean, there's a number of factors that lead into it. We are more flexible if it's an important investor that would add a lot of credibility to the story. Then we may consider, a 425, 450 US price range. But in terms of just turning it on, it would have to be on the back of some positive news that's pushing the price well above where it is right now.
Okay. A couple of questions about valuation as well. Any reason why the stock is trading where it's at, or do you have any comments on that?
Yeah, I think it's it's a struggle being a Canadian company competing with US peers. I think everyone knows we just listed on NASDAQ in August, so we're a relatively new story. We also don't have as many large US institutional shareholders backing us as some of our competitors do. So we've been working very hard to get the story in front of as many new investors as possible, establishing relationships with important banks that are very active in the space. So I think, just saw that we were at the Cowen and the Leerink conferences, and those are examples of great opportunities for us to get in front of new investors. And we need additional research coverage, public relations, build our team, our advisory board, and those are all things that we are working on. So, we hope that we will 6 months from now more accurately reflect where our peer companies are once all of these activities that are underway start to come to fruition. So it's a good time to buy now.
And which analysts are covering you right now?
So currently in Canada, we're covered by Mackey Research, Bloom Burton. And in the US, we are covered by H.C. Wainwright, Brookline, Maxim, Oppenheimer, and Zacks.
Okay, great. So we're about an hour's time. I'll let you guys have the last word. Maybe if you could summarize the value proposition, why you think investors should be taking a look at the stock. I agree. I think it's undervalued. I think there's some maybe misconceptions in the market, but I think this is a great long-term play. And we've seen with some of the transactions in this space that there's certainly a lot of value to be had here.
Yes, and I think we are, as a company, right in the correct technology platform. If you really look at the interleukin space, with the interleukin 4 or others that are being pursued, really an exciting space to be in. What we feel is that over the next 12, 18 months, we're expecting consistent amount of news flow coming out, whether it's partnering Mdna 55, Mdna 11 being in the clinic, clinical data for Mdna 11, or our next generation compound entering IND-enabling studies. So these are all very significant milestones that we're expecting. And in view of the fact that, as Liz pointed out, as we meet additional bankers, as we get institutions that are top-notch institutions to invest in Medicenna, that will drive the share price higher as we get more coverage from US banks. Again, that will spread out quite quickly in terms of, and I think our consistent effort to spread the story to new investors, I think that's important. But I think the core thesis around the company, namely targeting interleukins, is a space that is not going to go away anytime soon. And we have by far the best technology available to generate best-in-class therapeutics despite the competition. So we are very positive with the data we have so far and look forward to more data, particularly potential for partnering both Mdna 11, 55, and our pipeline as well.
Well, that's great. Well, congratulations on all the success so far, and looking forward to seeing what else you guys have in store for the rest of this year and going forward. Thank you, guys.
Thanks, Tina, for having us.